So it's my absolute pleasure and honor to introduce Dr. Parashurama. He is the CEO and co-founder of the Khufu Therapeutics, an early-stage IP-backed synthetic biology and organ engineering biotech. He's based in California. The company is focused on developing human livers from stem cells to treat patients with severe liver disease. He's a physician scientist, he's a bioengineer, former surgeon, professor, entrepreneur with training from MIT, Harvard, Stanford, UCSF, and other top institutions. His career is centered on liver biology, stem cell tissue engineering, organogenesis, and molecular imaging. Over the years, Dr. Parashurama has researched bioartificial livers and liver tissue engineering. He's trained in liver transplant surgery. He's earned his Ph.D. focused on liver differentiation from pluripotent stem cells. He's developed non-invasive imaging tools to study stem cell differentiation and living systems. Has completed advanced work in molecular imaging and early organ development, and he serves as the tenure track professor at SUNY, where he led a research group studying liver organogenesis and stem cell engineering. His lab-developed technologies include hepatic differentiation, liver organoid formation, organogenesis, and most importantly, computational/AI analysis of stem cells, aging, and cancer. Dr. Parashurama has a very strong academic record with 32 peer-reviewed publications, 3 conference proceedings, and dozens of technical and conference presentations. And he's an extensive student mentorship. He's founded Khufu Therapeutics in 2022 to bring that work into clinic and create new therapies for deadly liver disease. And we are truly honored to have Dr. Parashurama for this webinar. A few housekeeping points for the audience. Kindly please mute your devices and cell phones during it is going to be a 45-50 minute talk, and then we will have a Q&A session. So without further ado, Dr. Parashurama.
Thank you, Simon. And thank you, Josna, for invitation. This is very exciting to talk to the nuclear medicine folks. I know how kind of open the community is to biology, to science, to everything. I didn't put too many, I could have given an imaging talk today, but I talked more about our recent work and what the company's up to. And I think you guys will get some fresh new knowledge and ideas about what's happening in liver regenerative medicine. And you can see some of the potential for thinking about imaging tools and technologies to advance it as well. I'm just going to give an overview on liver regenerative medicine. I'm basically just going to talk about where the field is right now. I'm going to go through the different technologies and the clinical stages they're at, which I think I'm hoping is quite interesting to this audience. And then eventually, after that, I'll talk about our work at the company and what we've been up to. As I understand it, this seminar series is to honor Dr. Gambier. I am very excited to speak, and I'm very excited to have worked with Dr. Gambier. All I can say is, unless you know him personally or work with him, it's what you know is not necessarily 100%. He is an amazing person. I really feel we have to continue to celebrate him. His name should never be forgotten. What he was capable of and what he was doing was literally out of this world. I've put here 4 images of some of the work I did in his lab. I was very fortunate to do these different studies. On the top left here is a large animal study I did with cardiology and cardiovascular surgery. We used to do open heart surgery. We used to inject our stem cells were engineered with the reporter gene that Dr. Gambier had developed for many years, and it was optimized even more. So we had iron particles and a triple fusion reporter gene in the cells, and we were able to image them here with MRI and PET inside of a pig heart. The study showed the limit of detection of these reporters we estimated around 200, 300 million cells could be imaged with this technique. On the right is a totally different technique, bioluminescence. We were studying, we were trying to image stem cell differentiation. We had a dual reporter system. Actually, if you look at the bottom first, we would inject cells in the mice, the cells would die, and then they would start to grow rapidly. The reporter was for a unique different pluripotency reporter, which I'll talk about a little bit when we talk about human stem cells today. And what you could see is the reporter was on initially, turns off, and this was unexpected. At 72 hours for the reporter to turn back on. That would be not predicted by the field right now. So we found some new biology. Speaking of new biology, we did another study with intravital microscopy. And we used to, we did a lot of different things in this paper, including imaging cancer stem cell effects on the breast. In this case, we were studying the developing mouse, and you can see that at 3 weeks, we were able to image certain structures. We came back into the same mouse and imaged at 4.5 weeks. And you can see how the structures have grown. These are terminal end buds in the mammary gland in a fluorescent mouse. This is pseudocolored blue, but it's the same mouse. It's a green mouse. And using these simple techniques, we're able to study early organ development, mammary regeneration, and breast cancer, and how it interacts with the endogenous ducts. I could tell you, Dr. Gambier really loved this paper. He pushed for it to be on the cover of the Stem Cell Journal. The last one was a collaboration with electrical engineering, where we were building lasers and detectors on the same chip and developing these sensors. And we showed that they were just as sensitive as a bioluminescent camera. So these would cost $10. These machines, bioluminescents, cost, $500,000. I also wrote a tribute to Sam. I would suggest taking a look. The family really liked it. It was a little different angle than what a lot of people were writing. But there's been so many commemorative papers for Sam in his life, and I'm lucky that I was there during that time. So my area of expertise is regenerative medicine. There's already been a significant impact in medicine, whether you've noticed it or not in the clinic, and depends what you guys are seeing in a clinic. But the obvious things are things like organ transplantation, tissue regeneration. You may have encountered for skin or liver. Totally different area. All the increase in vitro fertilization procedures really goes back to regenerative medicine and stem cell research. The advances in cell therapy that we're seeing worldwide. Also tools for animals, which we're actually going to learn about today a little bit because I thought it's relevant. Transgenic animals, they're supposed to say cloning, chimeric animals, and even humanized animals. All are becoming possible because of regenerative medicine. The two areas that I became an expert in when I transitioned from medicine into more of an academic researcher is adult stem cell biology and pluripotent stem cell biology. So we'll be talking about these later in my session today. Now, we just said an overview on liver regenerative medicine, but the liver itself, liver as a field has been the leader in regenerative medicine. And that's what I'm going to talk about a little bit with you today. And the reason is because we're in the middle of a worldwide epidemic of liver disease. And so I think that's why it's becoming relevant. Liver transplantation, we mentioned, believe it or not, liver cell therapy developed already in the 80s and the 90s. In the 90s, there were clinical studies published. Organ recellularization, decellularization of a matrix, and recellularization. This is really a surgical technique that seemed a little bit silly at first, but it's actually being used in the clinic right now. First and foremost, I should have mentioned, of course, the liver regenerates, which is why it's a leader in regenerative medicine. But this, by the way, is acute regeneration in the chronically ill patient. Regeneration doesn't occur anymore, chronic disease. And that's an important detail that we'll try to come back to at the end. What's becoming very popular now is xenotransplantation. Kidney and liver are two of the biggest organs being addressed. We're going to mention this today, and it's something I had to learn when I have been working on the company the last year. Organ on a chip technologies to test drugs. Machine perfusion of liver. I remember showing this same picture 7, 8 years ago. It was kind of an idea. Now it's a really big thing in the surgical field where you can collect a donor organ and keep it alive on this machine at normal temperature. And finally, this bioartificial liver, which I worked on in college. And that is one that didn't quite work out. But the idea of putting something connected to the patient to support them, you'll see how that comes up in our talk today. So you can see that liver uniquely, maybe other than bone marrow, has moved ahead in the area of regenerative medicine with a lot of medical, surgical, and engineering type of techniques, and genetics as well with the pigs. So it's really a wide range of techniques now. And I do think there's a huge role for imaging scientists to play in some of the assessment of these processes and improving them. This is a slide that I show actually for my company presentation, just to show that liver, of course, has 500 known functions in the body, which is another whole discussion. But due to steatosis, fat, metabolic syndrome, alcohol, various viruses, aging, but also many other biliary diseases and autoimmune diseases can lead to fibrosis. 1 in 4 adults now has liver fibrosis, F0. 1 in 4 across the world. About 1/100th, 1/1000th develop cirrhosis. Once you get cirrhosis, it's irreversible. As you guys know, you're probably seeing it a lot in the clinic, requiring transplantation, or the patient's going to die. There's no other treatment over time. Unfortunately, we wait till the patient gets decompensated, very sick. At that point, their mortality rate ratio goes up, skyrockets. And usually that's when they get transplanted. But really, a cirrhotic liver, by definition, is irreversibly sick. There's no more acute regeneration. There's no more chronic regeneration from some of the stem cells. So it's done, and it needs to be removed. So we're going to talk about that today because there's 4 million patients with cirrhosis, and there's only 10,000 transplants in the US. In other countries, there's less. So you can see there's a huge need for liver therapies. I'll just mention also organ transplantation, we said, is the current treatment, but there's really a lot of limitations to organ transplantation. Some are pretty obvious, but if you really lay it out, you can see what all the problems are. There's not enough healthy livers. Most of the patients aren't even treated on the wait list. So a lot of them die on the wait list. When you harvest the organ, it's not alive for a long time. It also can get affected and viability can get worse. Operation is very expensive. Patients are immunosuppressed. There's really a wide range of problems. Now, we think we can wipe this out with our technology, but other companies I'm going to talk about first are also working on this. So there's a lot of things we can improve, even though liver transplant is the current treatment. So as I started the company and was working on this in my academic lab, which I've now left the university, I'm doing the company full time. I've had these ideas for 10, 15 years, But a lot of companies have now developed and got investments in the US and Canada. We're going to talk about mirror matrix and eGenesis today. We're going to talk about Ligenesis and satellite bio and Morphocell. And I think we're going to talk about Organox as well. So these are like three different approaches. You can use a whole organ approach for liver disease. You can use a cell-based approach. And these are kind of these technologies and machines that are becoming pretty useful and valuable. In fact, our competition, I've listed 5 companies here, are starting to enter the clinic, particularly Ligenesis and Miromatrix. So these are 2 companies I just showed you, and I'm going to talk about each one of these, but just to be aware, 2 of them are in phase 1 right now, or phase 1, phase 2. So this is going to be big news coming out for liver. And then this company has gone heavy into phase 1, phase 2, phase 3, eGenesis for xenotransplantation and for liver in phase 1. So it's really exciting time for trying to treat liver disease. Now, hopefully I'll convince you by the end of this talk that most of these companies are treating acute liver disease, and I don't believe they're going to work for chronic disease, but that's better than nothing. What I didn't show a slide, I thought I may have it in here, about the acute disease, because the cirrhosis is going up. Acute on chronic liver failure is going up. So patients with pre-existing cirrhosis are getting multi-organ failure, going to the ICU and dying in 30 days. That's the acute on chronic. Or they can just naturally decompensate and get regular chronic cirrhosis with decompensation. So these are all treating the acute disease right now. And some are obviously in preclinical development as well. Ligenesis is really interesting. The next few slides are going to be pretty relaxing, not too much science, and then I'll discuss some science slides at the end. They're using isolated hepatocytes from a donor liver. So this is allogeneic treatment. It's not a personalized treatment. Injecting them into a lymph node. So the founders of this company had shown 10, 15 years ago that you can grow cells inside of lymph nodes. The advantage is there's some space, there's some circulation, and there's a capsule. So they've shown that hepatocytes can grow inside of lymph nodes, and now they're doing this clinical trial where they're doing an endoscopic ultrasound injection into duodenal lymph node with hepatocytes. I don't remember where they're doing the clinical trial, but I'm pretty sure it's in the US. So the advantages of this is for acute disease. The reason I keep talking about acute and chronic is it's a really big factor. And honestly, I believe the investors in the liver business don't necessarily know the difference. In acute disease, when the liver is injured, it's secreting lots of factors that promote growth of the hepatocytes. So if I put hepatocytes in a lymph node in an acute regenerating, acutely injured person, these will receive the factors and start to grow. And I think that's why they're growing this well in the hepatocyte. These patients unfortunately need immunosuppression, but they don't need a big operation, which is something we're also revisiting as well. I'm not sure if the cells grow in chronic disease. And I do think later on, this is where imaging and liver doctors will come together, is let's say, okay, this patient survived this acute illness. How are you going to manage their liver, which is in a lymph node? Probably going to use imaging to see if the tissue's there. They'll have to do some blood tests, but to manage this tissue will not be simple. So I'm not sure if they can achieve the required mass. This is a big deal for us. I don't have a slide on it today, but a lot of times I begin this type of talk talking about how the liver is so massive It's a 500 billion cells, 500 billion. So cells have to, you have to achieve this type of mass to restore liver function. And there could be effects on the immune system when you're manipulating the lymph node, whether getting reactive to hepatocytes or tolerant to other cells. I'm not sure. But it's been approved right now because the patients are dying in the ICU. So the FDA will approve anything, unfortunately, as long as you can make the patient survive longer. Satellite Bio is a Boston company started by my friend of mine who's a pretty famous professor at MIT, and she's also an MD PhD in engineering department there. But she and also another colleague of hers has started this company, but they're not running the company. But the idea is they have found a way to expand hepatocytes, which is currently not possible. When we isolate them from a liver, that's the number we get. You can't expand them further. They found a way to expand it, but it's not published, so we don't know exactly how to do it. And then they inject into the spleen. So the spleen was a common preclinical location for liver cell therapy going back to the 80s. It started in Japan, actually, because the splenic vein helps form the portal vein. What happens is if you inject the cells in the spleen and the liver is acutely injured, the hepatocytes from the spleen can migrate towards the liver. I don't know if these guys want their cells to migrate, but it can happen. But also the circulation is shared. So the idea is if the hepatocytes function, it will go into the portal vein, it will go into the artery to the rest of the body, but into the or back into the spleen. But it'll go to the vein and then back to the liver, where some of these functions will be useful to prevent failure and also to promote regeneration. I guess I should mention that some of these solutions I'm mentioning, which have taken years to develop, they're not going to yet cure the disease. They're just going to temporarily support the patient so that their normal liver can grow back. So we'll see. We'll see if it stays longer or if it's really temporary. Um, I literally cut and paste the same issues. The same issues with the previous technology are similar to these. So I would say these are cell-based companies. And from our company point of view, we look at the cell as very fragile. And we also look at this as limited because they're allogeneic and you need a donor. So I think this is amazing right now. I just don't know if it's the future. It's the future. I think it's an immediate solution. I don't know if it's the future. There's a company called Morphocell, which has received a lot of hype recently here. It's in Canada and Montreal. So they're trying to add a lot of technologies together. They're actually using human stem cells, human pluripotent stem cells, which I'm going to talk about some more. It's also something we use in my company. They're making miniature organoids. An organoid is a collection of 1,000, 10,000, 50,000 cells that mimics the actual organ. And it's very valuable actually for drug screening and for studying organ development, drug screening, and disease modeling. And it's a very hot topic, something we're experts in the last 10 years in my research lab. So all of that is now coming to the company. So these organoids are put into a device with all these cavities in there. So the organoids can sit in these little cavities. And then this device, they gave very little information about it. But apparently what they're doing is they've encapsulated the cells to block immune cells from coming, to block antibodies from rejecting, and to allow nutrients and oxygen and toxins to enter. So metabolism and everything should be okay. And protein secretion should be okay because the liver makes 95% of the proteins in the blood. So apparently that works. I have some doubts about that, but they take this device and then they have it frozen and ready to go. So this would be in our therapeutic world. surgical world, it would be called a off-the-shelf solution. So they take this off-the-shelf solution, so it's out of a freezer, and they can go straight into a patient. And I found out that they're putting it into the peritoneal, underneath the peritoneum, just against the wall with a few stitches. And so they're not doing a big surgery, straight from the freezer, shipped, put it to the patient. And then the idea is that you temporarily support the liver. Apparently, it works without blood vessels. And then, so it's implanted in the cavity 48, 72 hours, and then replaces liver function and starts to function. And then it could be removed at any time. So that's the idea. So it really is a temporary solution, but the investors liked it. They don't even have a big article on their technology yet. Otherwise, I could have told you more about their technology. They're in Montreal, and kind of summarize what I mentioned here. They're reporting that they can function for 18 weeks in small and large animals, which I think is a really long time. Just, the problem with a material encapsulation as well as this foreign body is this can the body will produce a foreign body response to both the material and to the encapsulation materials. So I'm not sure how they can go 18 weeks because normally you get a foreign body response. And if this is the real design of the material, there's a lot of space that's missed because the liver mass is hard to copy. So I have my doubts about how this can copy the liver mass. It may have worked in animals. I don't know how it's going to work in humans. I also know about the organoid literature. We're actually studying their data right now, but we've studied 21 published papers, and I already know almost every organoid publication, the cells are immature. So maybe they did something with, especially with their organoids, but I cannot tell. So I'm concerned about the organoids. I'm concerned about the materials, and some of the design I think is not ideal, but it seems to be getting a lot of excitement in the US. Another one, which is, I mean, if you had invited me for this talk last year, I would not have even mentioned this. But I think it's getting very interesting. It's worth mentioning. When I was a young guy working at MIT, I used to do research at Mass General Hospital in Boston. So you just take the train over the river and do research there, which is now part of Harvard Med School, which I didn't know when I was younger, too young to know that. But there was always this transplant biology group there. I've been seeing them since I was a young kid. I didn't know what they were talking about. They wanted to do xenotransplants. But I think what's happened is with the advent of CRISPR technology, it's really helped this field. So currently, believe it or not, there's been a lot of optimization of transgenic optimization of pigs to make a universal donor for organ transplantation. The one I've listed here, I think, is the one that's probably got the best data and one of the most interesting ones. It's called this 3ko.7tg.ri, but it's got 69 gene edits. What I heard at the meeting is their latest pig has 187 gene edits. To me, that's absolutely, scientifically, to me, that's ridiculous. But when I looked at it, they have much better data with these genetically modified pigs than wild type pigs, or previous pigs used to have a few genes knocked out. So they've chosen this Yucatan pig. It's the similar size as human organs. You can get type O blood to prevent any blood reaction. The 3 glycan edits. So one of the issues is that we have preformed antigens. Sorry, we have preformed antibodies for a lot of these pig antigens. So if you put a pig in us, we reject it, hyperacute rejection within minutes. So based on that pathology studies, they started to remove, they figured out these might be the problem. And they just removed all these enzymes which transfer and build glycan chains on proteins. They've removed them all. The pigs were fine. And now it could be a donor. They've removed 59 pig endogenous retrovirus locations. So that counts as 59 gene edits. It's called PERV for pig endogenous, there's a spelling here, retroviral knockouts. And then they insert a gene cassette. I don't know everyone's molecular biology level here. That has 7 genes, human genes, into the pig that promote a healthier response. They prevent clotting through coagulation. They prevent innate immunity response. So they'll just allow things to go forward a little longer. And so far with these pigs, I guess, I don't remember when, but at some point in this field, they established the standard where you have to go from a pig to a non-human primate before you ever go to humans. And then non-human primates, they've gone 247 and 429 days. There's some that die earlier, but this is their best data so far, which is impressive. I would have never guessed that to be the case. I don't believe, I cannot say scientifically, but from other points of view, I don't believe putting the animal organ in the human is like a long-term thing. But temporarily, again, which is what I've been talking, a lot of these solutions are temporary to alleviate acute liver. For kidney, what's happening is they're putting this kidney in and the patient can get dialyzed, doesn't have to get dialyzed anymore, and they improve very quickly. And they've tested this in patients as well. And then there's other candidate pig genes that they're removing as well. So I think that's where they went up to 187 genes. So personally, I think it's quite interesting and quite impressive. Actually, it's a little bit relevant for our work. And this is another thing that my friends have been working on at Harvard Med School for many, many years, is trying to perfuse different organs in a machine. It's called machine perfusion. This company started in Britain in 2016. They actually have several papers. And what they've shown is, so you put the organ in this bucket, you connect to its vessels, and you just perfuse it oxygen containing blood-like substance or actual blood. You oxygenate the blood, you pump the blood, you can check the blood gases, and you can monitor this at normal temperatures, normothermia. So somewhere here is the temperature monitor. So I'm assuming they can cool as well, but they're trying to show that keeping at 37 degrees keeps the organ alive longer. So they have a lot of data showing improvements, less throwing out of the organs, more time for transplantation, shorten the wait list to transplant. So what happens is if you can get hooked up to one of these machines and you have liver, so if you have a liver failure and basically you don't have to wait for an organ as long, because as soon as the donor is available, it goes in the machine, it can be available, and then you can plan the surgery properly, and then there's reduced graft injury. So there's a lot of these benefits that are non-obvious from perfusing organs. So in other words, when there's an accident and there's an organ donor, you can go straight to these machines first, and then you can go to the patient within a few days. So it's extended that time. This one is really interesting because it combines a few technologies. So it is taking pig livers initially. It's a company called Miramatrix. This is the last one. And then we'll get into some of the work that our company does. It's taking pig livers, applying a process called decellularization, which I had in that liver slide earlier, where now you're just looking at the organ scaffold. There's no cells anymore. Then they're putting human hepatocytes at scale, the correct scale, 500 billion, injecting seeding into this pig liver scaffold. So now they have something that looks like a human hepatocytes in a pig scaffold, whatever that looks like, something like this. It's a recellularized scaffold. And at this point, that gets transferred into a bioartificial liver type device that I mentioned earlier. And now all of a sudden, you can support the patient, and this is happening and working right now. Miromatrix is in the clinic as well. So this gives you a wide range of these technologies. The four that are the three that are in the clinic is ligenesis, which is injecting into the duodenal lymph nodes. These transgenic pigs, that company is called eGenesis, and then this Miromatrix I just showed you have been tested on patients and apparently with some positive results. So things are changing for patients with severe liver disease, but we're thinking on a much larger scale. So I feel those companies are treating the acute liver disease, but the chronic liver disease is a severe chronic cirrhosis, 4 million patients. And if you look at the decompensated per year, it's 460,000. So even out of this 460,000, we're only treating 10,000 per year. We're nowhere near the limit for the chronic patients. We're nowhere near it, nowhere near. And now we have these temporary solutions. So we really need some more permanent solutions. The cirrhosis can be divided into the different types, pediatric, and some other types, but also viral, alcohol, fatty liver, roughly 1/3 of all the cirrhotics. Obviously, fatty liver has gone up significantly, and also these things can be combined in the patients, obviously. But in this case, some of these genetic liver diseases are places to enter and try your technology, which is what we've also been looking at. But what we do in our company, and it's based on over 10, 6 years, my PhD, professor for 9 years, can do the math, and also the last year and a half is the company. But basically, we can take induced pluripotent stem cells, which can be taken from an IPS, like a cell bank they have here in the US, where companies are just making iPS cells. Or we can take them from the patient. We can make for the patient. We have a patient with cirrhosis. We can collect their cells, make iPS. It takes 3 months, and it is expensive. That's one of the issues right now with personalized. But this would be the future for sure. So we can take these and we can grow the human liver. It sounds crazy. First of all, because I just told you the liver has 500 billion cells, but also we know in medicine there's architecture, there's structure, there's vasculature. So we are confident that we can overcome these hurdles by copying what happens during organ development. And today I'll give you a little bit the reason why to believe. I wasn't able to present all of our work. Some of it's trade secret, some of it's not published yet, but I have some data here to convince you guys that we can do it. So what we call it is kind of stem cell mediated organogenesis. This is a slide just on pluripotent stem cells. You know, I've been in this field 20 years, but I understand clinicians never have time to learn about this properly, including my own brother. So I don't blame you. But originally, we were taking blastocysts from fertilization clinics. And of course, it was easy to do in mice, growing these in a feeder layer and found that there's cells that stay alive. And those were called pluripotent stem cells. Embryonic stem cells also, but they were pluripotent. Pluripotent means they can make almost every cell and tissue in the body. And there was a lot of experiments you can do in mice where you can transplant back to the mice and see where the tissues went. I mean, it's a beautiful model for testing in mice, but if in humans, we can't put some marked stem cells into the embryo and put it in human. There's another technique that's very difficult called somatic nuclear cell transfer. I didn't want to get into it because we'll lose a little time, but it's a second technique, SCNT. This is used for cloning animals, by the way. And the third technique developed in 2006, where we can take any cell in the body, apply 4 factors. This is the Yamanaka factors to make iPS cells, and then turn those into any cell in the body. So this was a big deal. It's not because it's scientifically it worked. It was a big deal because it removed the ethical restrictions that people were placing on using embryonic stem cells, and it allowed the idea that you could make personalized stem cells. So this was a big discovery in 2006 while I was working on my PhD. And that's most commonly what, so just so you guys know, in human pluripotent stem cells right now, there's 100 clinical trials right now in the US. So it is active, internationally about 100 trials. I'll mention one other thing too. Usually in the first generation of these pluripotent stem cells was what's called the lineage specific differentiation, where you would take your cells, put them in the dish, you would apply a series of staged growth factors, 2 or 3 or 4 stages, to take the cells to a mature product, such as hepatocytes, such as retinal cells, such as certain neural subtypes, or even immune cells. So that is the first generation, and that those clinical trials are based on first generation stem cell technology. Now, we're talking about using a stem cells to build an organ. It's very different than what people are doing. But just to give you an idea, there are scientists trying to model the earliest organ stages, and there are organoids they've developed just for that. And what you notice if you look in the literature is these little discs. You put them in different places, they can actually replace liver function in a liver failure model. But when you come back, they have not grown at all. There's no growth. And I think that's why there's problem. This is another example from a completely different group. They put these organoids that they made in the culture dish using, in this case, it was a genetic technology to make this liver organoid, a pretty big organoid, and you come back and it hasn't really grown. But that's not what the liver does. The liver, as I'll convince you, grows very rapidly. So these were some of the questions we faced as we were starting our companies from a scientific point of view. How can we build a liver? But really, if you look in the body and even in mice, the liver grows very rapidly within a small, shorter amount of time, even in the fetal stages. That's when most of the growth occurs. It's not from a child to an adult, is not when most of the liver, it's going from the original fetus to the fetal liver. And then this brings up the question is, how is this growth aspect linked to maturation of the liver? There's so many complex genes, they need to be turned on properly. So is there some connection here? Why don't organoids grow? This is a bit of trade secret we have for the company, but I did just show you guys 2 examples of it. I kind of know why. I can't always share it publicly, unfortunately. But I'm going to share some of our data. And so this other technology language we've developed to think about how you go from stem cells to an organ. It's also based on a lot of developmental biology, which is not a popular topic with clinicians because we usually are facing very practical issues with patients' healthcare. But because I did a PhD, I had the fortune of being able to dive deeply into embryology and developmental biology, learned on my own to understand this problem. And then ultimately, how can we reverse the chronic liver disease? So I just put this slide together just to show you about the types of things that we had to look at to understand what's going on. we kind of know now how this gut tube forms. It's known in science. It's known by us. I became an expert in this during my PhD. I was a general surgeon. I obviously got interested in the GI tract and the development of it. There's early factors that can control the initiation of the entire liver. That's this. There's other factors that control cells that migrate onto the mesenchyme. And there's other factors that remature. So here, they're kind of low, but they turn on at 10.5. These numbers are during organ development in the mouse. So the mouse, E8.5 is when the organs just form. 9.5, they're just starting to mature. So that's why we've shown 9.5 across here and 10.5. So now you have a little liver structure here that's beginning to grow. So we think that these steps are missing in the current differentiation protocols. This is a lineage map that, people have been using this since the year 2000. I know that because I was in the field starting in 2001. So, and I know the literature, original literature. So this is what people think, even really smart people think. We go from stem cells, we go through some gut steps. Then once we're committed to liver, it's all about maturation towards mature hepatocytes. However, what we think is this is somewhat wrong. And we found there's actually two missing stages in liver development. We're less focused on this one right now, but we became experts in this one. So we are probably the national or international experts in these cells. These are the ones that start to migrate out into the mesenchyme, and they're pretty much the first cells that lay down the liver architecture. So we think they're very critical for what we're trying to do, but they're ignored in the entire field. So we have an entire 2 papers coming out on this, where we showed that these cells are really interesting. Now it's a little more bioinformatics, which again, I know is something maybe our audience may or may not be familiar with from being a clinician every day. New topic, obviously, but if you just look at these colored charts here, these pie charts, only thing I'd want you to notice is this brown slice here and the yellow. So as you go from this MHB cell, which is at 9.5, to the hepatoblast at 10.5, which is now you're on a maturation stage, you can see the switch from signaling to metabolism. So there's a huge switching mechanism here. So these cells are migrating, laying down the liver architecture, proliferating, because there's other evidence like the red, which is the cell cycle, they're turning on, and then they switch, and now they're activating metabolism, and that trend continues all the way from 10.5 all the way to the end. So usually we take it out to 18.5 is like the oldest fetus we can get before they're born. In this case, we've shown 16.5. So we're showing here a maturity classification. So there's a switching mechanism that turns on maturity. You can see it right here. We even know the factors. And these molecules is something me and my team are very familiar with. In fact, probably experts with so many of these transcription factors that control the switching. And we're also now experts in some of these signaling pathways that turn on, that switch on and off from here. These are up in the migrating cells because we said they're high in signaling. So we think these cells are critical. I'll show you a little more data why we think that is true. But to answer one of the other questions we had is, how does the liver get so big in the first place? So what we did is we took online data, we did 3D reconstructions of tissue that was sliced at 4 millimeters, and they were histology sections that were cleared, so you can reconstruct the data and reconstruct your organ and segment out the rest of the tissue. My students did a great job on this. And what started as like a simple master's project, we actually discovered quite a bit of things that nobody else knows in our field. One of the first things, I mean, I've listed out a little bit here what we discovered, but we applied some mathematical modeling to show we could quantify the liver growth in mice, but we also did in humans. So now I have exactly how fast the mouse liver grows and the human liver grows. And what you see in the mouse is it goes 10 to the 5 or 10 to the 6 fold increase in mass only in about this 8.5 to 18, 10 days. So we've been focused on this 10 to the 6 fold idea of change in mass. So in the humans, it goes longer, but if you look at 10 to the 6 fold, it's about 60, 70 days. So we think if we can get 10 to the 6 fold change in our tissue mass, we'd be far ahead of anybody towards making a new liver tissue. So a few other things we discovered from this imaging study, which started pretty simple. So then, since we're interested in these migrating hepatoblasts, we actually made organoid models to form those migrating cells. This is the basis of the organoid that we form up to on day 14, we form these organoids, and then we take them out for 2 to 3 days. But we showed that by changing the conditions, changing the biology, we can make these migrating cells. And these migrating cells we were not expecting are very similar genetically, transcriptionally to the ones that I just showed you in mouse. So we're the first group to actually make these migrating cells. On top of this, this is our human data now. We created a mapping system where you can follow the trajectory from human stem cells to gut tube. So if you're missing this migrating step, you kind of jump into the middle here. But if you keep the migrating step, you go up on this line. And now we can see as the cells mature towards fetal hepatocytes, they need to travel on this line, and then they need to travel again towards adult. And what we've shown is we figured out how to switch these cells from this place all the way close to the adults. Better than anyone else right now in the field. Using these pretty simple techniques, we were able to first, when we measured just 50 liver genes, we were 2 of the top 5 out of all papers published. And we had a very different technique than anyone else. We did not rely on any of the stuff people are doing. We relied only on these ideas about building out these migrating cells. We had the best, when we looked more at 1400 genes, we basically had the best cells for gene activation out of the top papers in the field, and the right controls here. So this is a picture of some of the tissues we can generate. We have 2 models. There's one called the iLiver I showed in the beginning, which is personalized, and then there's this A-liver. So the iLiver could never be done with humans. So this is mouse iLiver in mouse. We showed that we can make tissues pretty comparable to the size of the mouse liver. And we could take this much farther in the company. And if you take these tissues and we measure the rate of their growth, we're on an exponential curve going in this direction. So we think we can make massive amounts of liver, 10 to the 6-fold liver, in only 1 to 2 months. We've done many of these studies in different models and found lots of interesting information about our tissues. Obviously we have liver markers positive in our tissues. We've also taken these cells. I know we're running a little short on time. We've taken these cells, we've put them in devices and showed we can reverse liver failure. So they're capable of reversing acute failure. And we are experts in building human livers. These are some of the technologies we need to grow those livers. We have them under patent. We have them in some of our publications, and we have some trade secret on these as well. And our vision really is to not only go for liver, but start to go for some other tissues inside the body whether it's tissue rejuvenation or replacing an illness. Obviously, anti-aging research is really huge right now in the US. And so our technology could be thought of as a rejuvenation technique as well. But really, what we want to do is treat chronic liver failure. And in general, what I told you in this section, after we went over the existing technologies in liver, I went over some of the stuff we've done in our group. I mentioned the imaging study where we studied growth quantitatively, and then also we became experts at the gut cells and also those migrating cells. We lay down the architecture, and then we can build these little tissues, as you've seen. And then we're moving forward. We have a lot bigger plans now since I started the company, and that's about it. I appreciate the invitation. As I mentioned earlier, this is our team, my co-founder Kitana, and computer team, scientific and business advisors. I believe Paul Morgan spoke to this group earlier this year. We're good friends. And then these are like many of the students who helped out when I was at the university, many of our old students. And I'd be happy to take any questions if there's time.
Thank you very much. This is Raghu Halkar from Emory.
Hi.
An excellent talk and vision in the future. So, nuclear medicine has evaluated liver function with hepatobiliary imaging with immunodiastic acid that assess the hepatocytes and Khufu cell function with technetium sulfur colloid. Having said that,
is it technetium? Technetium's used for liver?
Technetium labeled compounds, immunodiastic.
Using SPECT. So which is the I don't know the most common one for so I would have thought you guys are using even FDG for some liver.
It goes to the liver, but we don't use it for liver functional assessment as such. But 90% of the radiopharmaceutical that we use end up in the liver, whether it is
exactly.
And many of them have hepatobiliary excretion, like somatostatin receptor agents like dotatate. Comes to the hepatobiliary system. FES, the estrogen, estradiol. The only few traces that do not go to the liver are technetium MDP. I can count on my fingers how many traces do not go to the liver. So liver probably is a very
organ of interest to you.
Diverse organ and attracts diverse molecules as part of the detoxification process. I-131 doesn't go to liver, but
which one?
Later on it goes to the liver for detoxification. So my question would be, maybe once your technology matures and when you get the liver in place, then many of the radionuclide techniques that we already exist and can be used to assess different aspects of the liver function.
Liver tox and liver function. I agree. I just think that that's the simple way we think about our company. We just want to make more livers, And I believe that's the future. That's the application. That's the correct application.
It's an important requirement right now, certainly.
With the advanced liver disease right now. But obviously for biopharma. So for hey Deepak, how are you?
Good to see you again, Natesh.
If you return some of my messages, we'll catch up soon.
Yeah.
We want to make livers, but also obviously for biopharma. So there could be some for pet development of some of the tracers you guys develop. There could be ways we can integrate these tissues, but you can see what the competitors are doing. You can see that now the investors are interested in this. It's a major public health problem. Finally, the liver is getting attention, even though It's a hugely ignored organ in medicine. We also have another project we're actually trying to develop better ways to monitor. The test we use for liver right now is almost embarrassing that we're using a leakage of an AST as a way to assess liver function. The liver has 500 known functions.
Yeah.
Just to give you an idea, when you eat a meal with amino acids, the liver can normalize all the ratios and spits it out as a constant ratio. So there's so many things we can do with developing improved liver function tests. We're also working on at the company. And yeah, that's a great point. I didn't mention biopharma as much for building our livers, but that's obviously of interest.
I have
Hi,
one interesting question. Sorry, I'll take 1 minute.
Please,
Dr.
I
Parashurama.
can stay on as long as you guys want to stay on.
Yeah, I want to know two things. They are not completely relevant. Do stem cells live forever? For a question for Parashurama
Yes,
is an important
actually, I
question.
have. So they've shown that the hematopoietic stem cells that self-renew in your bone marrow can far outlive the age of the organism. In other words, they can keep transplanting in mouse after mouse way longer than after the 2 years that the mouse lived. The several months that the mouse lived. So yes, the answer is any cell that's self-renew, even these pluripotent stem cells I didn't mention because I didn't want to get too much in the biology, but they self-renew infinitely. So that's why we can make unlimited livers in this company. If we get the cost, little things lined up. I have extra slides I can show you, but yeah, that's correct. They do live forever. It's shown in the bone marrow, and our stem cells, pluripotent stem cells, also can indefinitely grow.
Thank you.
Hi,
Good question.
This is Jyotsna. Hi. That was an excellent talk. I just wanted to know how expensive this technology is. I mean, to have it in India, is it possible? Like, we see so many cases that patients who need liver transplant.
You're going to see more and more. It's not going to go away. I don't even know if the obesity drugs are going to solve all these problems. My cousin died of cirrhosis. He was in his 40s. So, yeah, I mean, I thought about all these things. So what we really want to do, I also didn't put the slide in, is we want to inject a small number of cells inside of a patient with severe liver disease and chronic liver disease, and have the liver grow like a new plant. So in that case, we would avoid surgery. It would be very inexpensive. You would not need a surgeon. You would just need an ultrasound guided injection or something. So any doctor can do it in any hospital. So, or, an interventional doctor can do it. So we are thinking about costs, obviously. And I'm learning too, though, that business people, they do not actually want to cure a disease. They want to keep the patient sick. I can't even use the word cure when I talk to business people. It's ridiculous. And I'm also worried if we really got things working well in large animals. So when we can grow a liver in a large animal, I'll be jumping up and down. That's some of the next milestones for us. Then we can talk about how to make it cheap and things. So obviously it's been on my mind from the beginning, but I had to get rid of my final idea to the initial idea so that the business people would believe it. Like, we didn't even want to treat acute disease initially. I wanted to go after the chronic disease, but we have to do acute disease just like these other companies. I can't even prevent it. So we have to make a liver, then I have to transplant into the patient and show our liver tissue and show that we can reverse the disease. So I have to start on that, but we have bigger ideas if we can get going and get enough money.
Yeah. So maybe another few years, if any of us can collaborate with you, that would be lovely. I mean,
yes, we're definitely open to collaboration. I've been talking about it recently with some people and getting on some grants and things like that. So absolutely, I just need NIH to fix up their funding a little bit. And yeah, obviously, if you guys are anyone down in San Diego, I'd be happy to meet anytime. Anything from this conversation was interesting today. Thank you for everyone getting up early or late in India. Deepak as well. Thank you.
Hey,
Gotta
Thanks,
Look at the
Natesh.
question.
Sorry, yeah, a wonderful presentation. Thank you so much. And you've come a long way. We haven't been connected in a long time, so
I
I
know, yeah.
hadn't realized how far you've really come. And I'd love to dive deeper, understand what things are. First of all, I
Yes.
think we're all rooting for something like this, so something to sort of help with the HCC and acute and chronic both. I mean, so I can drink more.
Oh, I didn't even mention ACC. Yeah, exactly. So you can have a drink and not worry about it.
Yeah, I can drink and not worry about it. But also, I mean, in general, I think this is certainly definitely significant problem to be addressed. I'd love to hear more. I don't know if this is the right forum, but in general, overall, maybe we can chat up sometime.
Of
I'd
course.
love to hear more about what are you facing in terms of getting it into the clinic and into the next stages and the following stages.
Yeah, I'd be happy to. You're up in Bay still, or where are you these days?
I'm still in the Bay Area, yes.
Okay. So I'm down in San Diego. I decided to do the company down here, but I'm up in Bay, and I'll track you down. I'm actually there next week, but if not, I'll
Oh,
be
brilliant.
there every month. I'm there for about a week, so
Excellent. Excellent.
catch up. Stephanie's down here, by the way.
I know.
Yeah,
Yeah,
yeah.
I'm aware of it. Yes.
But yeah, I think it's important to keep Dr. Gambhir's name alive. I don't know how Deepak feels, but I'm pretty sure if he was a regular American, they would have made a movie about him already, and there would be 100 books on him. That's how big he was. It's hard for people to even conceive of it unless you work right next to him and go into his office and meet this person and see them every day. I really insist on this that he should, every Indian kid should learn about Dr. Gambhir, honestly, who grows up.
So to echo what
They
you're saying, Dr. Prashoma, actually the IASNM MISI webinars are dedicated to Sam Gambhir.
right. We have to keep his name alive because what I noticed in America is people will drop his name because he's, slowly forget him because he's not American. So
And
yeah,
I
it is our job.
office sessions at the national conferences as well. I think we talk a lot about, and you are absolutely right. We talk a lot about all the, people from the Western world or the European world, but not Sam,
We have to keep his name alive. I mean, I wasn't best friends with Sam. I mean, even at the end, we were arguing a little bit, but that doesn't matter. But how much he had done and accomplished, it's incredible. I mean, it's unthinkable for any person. It's unthinkable what he was doing. He was
way ahead of his times. He was born.
not even that. He was so talented. I can't even explain it. Within a year of being there, I used to tell my friends in Boston that he could run the entire country with one finger. He could run the entire government with one of his fingers. That's crazy. He probably could. Do you guys know that he was offered the Dean of Harvard Med School? A lot of people don't know this stuff. That's like the biggest position probably in the world in medicine, in my opinion. Harvard, if you've ever been there, go there. You'll just see it's such a big thing. I grew up in Boston, so I know. So he turned down he was offered the Dean of Harvard Med School. I think that's a really big deal that a lot of people don't know, and he didn't advertise it, but he turned down the position. So that's when the the top people in Boston got to know him because he had applied for that job, and then they were so impressed with him too, they gave him the job. So yeah, just to feel when it happened, I was in shock. I saved every single video that was online for him. So I have a list of 20 videos. Would Sam ever want to get inspired, watch one of his videos? And then I was lucky enough to write this little memorandum in one of the, not in Nature or Science, just in a regular journal. So one of the guys invited me, and that was very, what's the term, therapeutic. But the family liked the one that I wrote. They contacted me after. They said, I would really like to. So,
If I may just add a word, actually, our very society was formed because of SAM, the Molecular Imaging Society of India.
right.
And we're trying our best to keep his name alive. And actually, we were fortunate to have him in Hyderabad, where I organized
nice.
a couple of lectures, one in Usmania University, one in my hospital, and one at CCMB. So we were lucky to have him. And also, we actually used to publish a newsletter online. And our first newsletter we dedicated to him. And actually, recently I visited Stanford. And I was so happy to visit the department and
Oh,
see
nice.
the inspiration wall that they made. So
Yeah,
for
they
me,
have
Sam,
that stuff, but I just meant, I didn't mean in a negative way, but I felt like because there's so many famous people in Stanford that it can easily be forgotten. There's so many famous people there in Stanford. There's so many big professors, maybe not necessarily in the radio, just like throughout the university. That's why I also felt it that the Indian community, they had a different version of Sam than some of the other guys. I just felt it immediately when it happened too. I noticed right away. Just
What I think, Dr. Parashurama, I think there is no way that the memory of Sam will fade. People like you will
not,
carry the baton. The lives that he touched, I think, will carry on the mission. And
yes.
I think he was fortunate to work with people like you. You were there. Jyotsna is all
Very
praised,
lucky.
and there are so many who, whose paths crossed with Sam.
This is what I noticed, that a lot of people, because he got so much tragedy in his life at the end. So then everybody started talking about that, how he was dealing with the tragedy, how he was handling it. Okay, but I got to work with him before the tragedy. So when I wrote the review, I wrote just about that. I didn't write about any of the negative stuff. So that's negative. So that's what people have forgot just now, how high he was going before all this stuff happened. It was crazy. It was absolutely insane. The ideas that he had, none of us got enough time with him. We always wanted more time with him. And honestly, yeah, it was crazy to work at his lab. It was like a madhouse. But it doesn't matter, just to get some exposure to a person like this. I mean, I gambled a lot of my career to come to work with him, but I really wanted to see who this person was, and it was incredible. If you really work with him, and you go to the conference and everyone was always trying to chase him down. That was always funny. But yeah, so either way, it's been a pleasure. My talk doesn't, we've gotten a little more into therapy, but I do know the diagnostics and all the fundamentals I learned as lab are going to come up for our company as well.
Well, thank you very much for this very insightful talk. And that's the whole spirit of these webinars, that we want to bring in more physician scientists to learn more, not just the nuclear medicine and the diagnostic aspect.
Yeah, I know this was not very atypical.
Yeah, and that's what we really like.
Developmental biology, genetics, surgery in this talk today. I apologize, but hopefully it was a little interesting.
That's why we invited you. We wanted to learn all those about, and
Yeah, I know, but that's the beauty of the nuclear medicine community, is that it's a really open group. I found that to be pretty amazing, actually, when I was there.
Thank you very much for your time, and
Yes.
truly enjoyed the talk. And stay tuned for next month's webinar. And
Yeah, I'd be happy to join in next time.
Absolutely. Absolutely.
I'd like to thank you, Natesh, again, personally.
Thanks, yeah. No, thank you. It forced me to get some of these companies together and stuff. So it was good for me to know all this stuff. So I appreciate the opportunity. And it's the first academic thing I've done since I started the company. So.
All right. I wish everyone a very nice rest of the day, and we'll meet again.
Sounds good.
Next month. Bye.
I'll message you, okay?
Yes, thank you.
Bye.
Okay.
Bye, Natesh.
Thank you.
Bye, guys. Thanks a lot.